The overall goal of this procedure is the biochemical and structural characterization of a carbohydrate substrate-binding protein from Streptococcus pneumoniae.
This method can help provide key data to aid in structural studies of substrate-binding proteins, such as the identification of their buffer stability profiles, oligomerization states, and atomic structures.
The advantages of this procedure is that it provides data that can increase the success rate of crystallization and structure solution for substrate-binding proteins in a robust and straightforward way.
First, prepare 48 buffer solutions with a pH range of 4.0 to 9.5 and sodium chloride concentrations ranging from 0 to 0.5 molar. Dispense 40 microliters of each solution into a 96-well plate. Obtain a purified solution of the chosen substrate-binding protein. Add 5 microliters of the SBP solution to each well for a concentration of 1 to 2 milligrams per milliliter. Then, add 5 microliters of a 20 times concentrated fluorescent stain to each well. Mix the solutions by pipetting up and down. Seal the plate with transparent adhesive film. Centrifuge the plate at 112 times G at room temperature for 2 minutes. Configure a real-time PCR system to ramp from 4 to 99 degrees Celsius at a rate of 3 degrees per minute with a 10-second hold time. Set the system to take fluorescence readings every half degree. Set the fluorescence filter setting and assign the samples.